Hello, everyone. This is Songwoo Kim from the Department of Biomedical System Informatics. Today, I will tell you about how to detect mismatch of samples caused by a person's mistake effectively in the clinical training stage using NGS. Furthermore, I will talk about new technologies that can correct this. Currently, precision medicine is mostly genetic-based, as you can see it in the picture. Modern precision medicine is using the way of examine genes and gene variants that every patients have individually and applying the optimal treatment method according to the kinds of variant detected to increase the viability of patients, whereas patients with the same disease were treated with same treatment in the past. The technology who plays the most important role in this process is next-generation sequencing. This technology can detect gene aberration by examining multiple genes in a disease sample that a patient have a problem with, and this could make us able to realize precision medicine. These kind of precision medicine is applied on cancers the most, as you can see in this picture. Finding gene aberration in cancers and treating patients with the optimal treatment according to this aberration is actively conducted. For this, a sample-derived tumor and a sample that is not cancer, generally a pair of sample from blood or saliva, can effectively detect somatic cells in cancers, so two samples would be obtained from one patient. We test gene test using next-generation sequencing with the two samples, and after analyzing gene aberration we obtained from it in bioinformatics ways, we can acquire a report to a doctor that can be applied to various medicines and clinical trials which can be used for the corresponding aberration, as you can see in the right picture. Therefore, we always have a pair of pairs per patient. In other words, the tumor and non-tumor parts must be obtained and proceeded. It is true that great amount of errors are occurring in the process of examining these samples. The picture on the right shows the most common examples of errors. These errors has an accuracy limit of detection that NGS technology has, and various problems like contamination and damage on DNA that can be obtained from other times besides when conducting NGS on samples. We can prevent these situations, but a contamination of sample is most effective once it happens. There are roughly three kinds of contamination of sample. One is intertumoral contamination. It's when other tumor cells contaminate each other when there are many cells. The other one is called intratumoral contamination, and it happens when other people's cells get mixed in with someone's cell. Lastly, when samples get mixed up when NGS test is conducted on many patients and acquire many samples, we call the sample mix-up. Before the presentation, we have developed a technology that can detect contamination in NGS effectively and correct the contamination. This was a technology that can detect vector contamination almost 100%, and we have obtained a patent and transferred technology. Now, I'd like to tell you about sample mix-up. That is when samples get mixed up in this presentation. In this case, multiple patients, generally when it's a NGS clinical test, we target few genes. And it's common to conduct sequencing at the same time, instead of one person at a time for the efficient expense. For the correct interpretation of the result, at least one tumor from a patient should pair with one matching control group sample exactly. When it gets mixed up and conduct analyzing one patient's tumor with other patient's blood sample like they are control group to each other, a huge error can occur. In this case, the variant of reproductive cells obtained because they are two different people can be detected, like they are somatic cells, and it can be misinterpreted like there are hundreds and thousands of somatic cells. For a patient, this can be a great disaster when he or she went through NGS clinical trial to get the optimal treatment, and it was failed to find correct gene variants and lose the opportunity of getting the optimal treatment. People have been asking me if these kinds of situations really happen. We have been conducting various NGS tests for around 7-8 years, and when you look at the results, these happen very often. The reason why people don't know it's happening often is because they don't know about how the results are detected. We have seen these kinds of cases a lot, and according to the thesis reported, sample mix-up happens in international consortium large-scale project that goes through quality control. CQ, which is a lot more precise than NGS clinical trial conducted by organizations. You can see on the bottom in the middle. This shows sample mix-up found in International Consortium Cancer Tumor Dielectric Analyze Consortium. The right one shows sample mix-up found in a project called ENG. Considering that these kinds of things happen in a project where through QC is conducted, I can say that these actually happen a lot in our daily lives than we think. We were not the first one who detected this problem, and reports have been reported. There are a few technologies that try to correct this problem by detecting sample mix-up through analyzing NGS data. What you can see on the left is a technology that was developed in 2017 by Broad Institute called NGS Checkmate, and it was reported to Nucleic Acids Research. In the middle is a technology called mixup developed in Adelaide University, Australia. The one on the right is compared developed by New York Genome Center. From our experiences testing all these technologies, they work greatly for whole exome sequencing that we conduct for studies, that is large-scale sequencing, that sequence the whole dielectric or coding genome. However, when we conducted it on a NGS panel where NGS trial test is actually conducted, by which I mean a platform that sequence only few selected genes, the accuracy was quite poor. The reason for this is because we detect with SNP, different reproductive cell variant that different people have as a way to detect sample mix-up, and fewer the genes that take in NGS platform, the fewer SNP, and the accuracy can be poor according to this. Therefore, we have developed a new methodology called BAMX checker to develop QC, that is routine in NGS trial test. The biggest difference of BAMX checker is that it maintains its accuracy even with gene NGS panel that uses a few genes. The most discriminative technology we tried to develop to maintain accuracy is that we made priorities by making the optimal list of SNP that we can utilize first from a large-scale data, and when there's a new gene panel. We developed an algorithm that can reorganize gene SNPs on the spot that can be utilized according to this. These SNP lists reorganized on the spot can have a degrading ability that can distinguish people more efficiently than SNP lists previously fixed even with few genes. Accuracy analysis was conducted on this algorithm, and this is the picture of analyzing the accuracy of the data of various NGS gene platform on the left. The one on the left is a picture of whole exome sequencing. RNA sequencing when sequenced the whole coding area, when sequenced the whole transcribe, and when sequenced both whole exome sequencing and RNA at the same time. There are a large-scale panel with many genes and a small-scale panel with few genes out of all the panels, and we have tested all kinds of panels. When we tested, there was a whole exome sequencing that sequenced the entire coding area, and the RNA sequencing that sequences the whole transcribe. All of those three previewed methodologies could detect the differences among the people who have been mixed by mistake with almost 100% accuracy using our technology. However, we could find that the smaller the size of genome we test, the lower the accuracy of the previous technology. The panel you see at the right bottom, which was utilized in an actual clinical test, was a very practical case selecting only about 40 genes for sequencing to make it cost-effective. These cases, shown as red-colored BAMX checker, could be detected with 100% accuracy using our methodology, while we could find other methodologies showed the accuracy of 340% at the lowest, 780% at the highest. As said before, if the accuracy becomes low like this, the patients who participated in the test will be endangered. They will not be able to get an ideal medical treatment then, and I think we should not let any kind of this things happen. We are insisting that we must always conduct a test first. before doing any test using technology with 100% accuracy to identify if this kind of thing has happened to us or not. While developing this algorithm, we analyzed some more things to make our work more practical. One of them is that, if the analyzing itself, when it comes to utilizing NGS clinic requires hand-on time, which means that a man needs to physically be there to make an analysis. And I also minded that each analysis preparation time could be a burden on an actual pipeline. I figured out that therefore, we should maximize automation of work so that with given data only, we can have automatically analyzed one on our hand and print out the result without doing any further tasks on our own, so that it can be used practically. And the thing we developed to make this happen is like, when we conduct NGS test extracting tumor and blood from several patients, there will be information, so-called annotation, that includes patient's tumor, normal, patient number, etc. attached to the file name. Since the information can vary depending on the timeline, as well as on what government agency in charge is, we have to make sure to have a whole picture of the situation whenever conducting each test. To solve this, when given a file list, we use Shannon entropy and information analysis method, by which we can automatically identify and know if there are any variants and automatically extract the information they have. Also, if just a file list and data are given, it can enable us to detect the existence of mismatch by viewing the patterns written on the list without any further annotation works. Therefore, the hand-on time could be almost gone actually, and we could just routinely check and move on to the next step. The last important thing is a running time. It takes a very long time to make an analysis. If a massive converting material is required, it will be very difficult to be utilized. When we were developing the method, we used a way of selecting an ideal list and finding out a solution within the list only. The required calculation process is much less than that of other algorithms. What you can see on the left side is an analysis of the running time. The two orange-red ones on the very top are the results of our BAMX checker. Since BAMX checker was developed to utilize multi-core, which uses several CPUs, when on the running mode, it can analysis data quicker than any other pre-existed technologies. As you can see in the middle, the other analyzing tools, which were previously precise, need a very long time for the process. We have technology and accuracy that can minimize a burden in terms of time and labor when we actually start operating the process. To sum up, our technology is about precision medical technology, which is playing a key role in NGS, clinical examination, followed by our development of an algorithm that can prevent correct, unwanted mixture of samples of patients. As said before, even one single error were to occur to a patient, it could bring him or her such a critical result. Therefore, it is no exaggeration to say that we have to be always 100% accurate. Also, if we look at the process of NGS clinical examination, normally a pipeline is composed. So when there is input data, it is standardized to acquire a result value according to a certain protocol. Therefore, we can draw a conclusion that depending on whether or not we efficiently put errors in a standardized pipeline so that we can move to the next step without any further care, there would be a difference in the practicality. Aside from being accurate, we can minimize hand-on time, and we can even reduce the running time. So, we can say we have now all the tools we need in order to make a routinely standardized analysis. Therefore, if we use the routine pipeline well, we will be able to ensure the accuracy in NGS clinical examination research. Also, it will make our patients give us credit. This algorithm was introduced in Bioinformatics Journal, which is one of the top journals in the bioinformatics field, in 2019. It obtained a patent in Korea, and now it is in the middle of the process of PCT application. Thank you very much for listening.